Welcome for those who just joined us on the best booth ever, of course, of VivaTech here at Sanofi. We're very excited to share innovations with you. You're already here. We will talk about LLMs, large language models. See, this time is a good one. In research and development, search and design in chemical spaces. What is it exactly and how does it change the game? We have the chance to have you with us, Sven Jager. So you will talk about that together as your data R&D, let's put it that way, R&D and Computational Science Manager at Interim. So let's start with the first question about LLMs, as we say when we're best friends with large language models. What is it exactly?
Hello. Yeah, perfect. Welcome, everybody, and thank you, Christina. It's an honor to speak here at VivaTech, and also about one of my favorite topics, large language models, and also what we developed or what we cooked at Sanofi. And but starting with the basics, and let's explain you what large language model is briefly. First of all, I'm very happy living in times like this, because I don't need to start really explaining the large language models, because everybody experiencing large language models also in their day-to-day life. We are using ChatGPT at home. When you have the honor working for Sanofi, you can use Concierge actually, which is, let's say, our own language model assistant that we have that helps the Sanofians for the day-to-day work. So LLMs are already a part of our life. But technically speaking, what is an LLM? An LLM, you can envision this as a huge neural network. It is inspired from the neural network that everybody has in their brain, but this is a specific neural network that's called transformer, or most likely transformer. Why? Because we are kind of transforming sentences. The LLM, that means like the large language model, want to, and specifically when you're talking about architectures like GPT, wants to complete a sentence with text or want to exchange the missing word in a given sentence. And this is mainly the language model itself. So it learns on language, it learns grammar, it learns kind of the semantics that we are using. And that's the typical text-based large language models. On the other hand, we have also different language models in Sanofi. We are a pharma company. We are very innovative. So we are also working with language models that does not learn on text. We're working with language models that are considered as expert AI, that are working on natural sequences, like protein sequences, RNA sequences, small molecules, really taking kind of a different language into account. And these are the two language models we have so far.
So tell me, what's the difference between text LLMs and also molecular LLMs?
The difference between text LLMs and molecular LLMs is mainly the training data that you have. The architectures of the model are the same. Maybe you are tweaking a little bit the pre-training tasks, but typically you are replacing words with amino acids when it's a protein. You are replacing words or sentences with a small molecule where the words are the functional groups of the molecule. And so you have a kind of a similar context actually that you're using in these models. So it's mainly the training data and the process and how you are using it. How you are using it. And that's the good thing actually. You have the text-based models. It's cool, you can chat with them. You can directly interact with them. You have a clear application. The expert AI models, you cannot chat with them, but they can generate molecules that are innovative. So they are technically very smart, but not conversational. And vice versa, you have the text-based LLM, which is great to have a discussion, which is great helping you with your emails or your tax returns, but it's a very bad scientist. And that's kind of the thing, so you have these two things.
So tell me, how do you use LLMs in the drug design process?
So in order to explain this, I need to explain a bit what is the drug design process and what are the challenges actually in drug design. Of course, in Sanofi, we're aiming to moving forward with a digital AI-based and quantitative platform to predict, because we need this also. Why do we need this? because the drug design problem is very, very difficult. Typically, you have a target like this. This is a protein structure. Then you want to find actually, for example, here a small molecule that binds this structure. This is already a super difficult task because the solution space of these man-made molecules is huge. How are you tackling this as a chemist? You have a lot of experience. You worked on this on your university, on your doctorate, on your postdoc, and so you have trained your neural network a lot. But you are still a human. You are challenged by complexity. And the space we are talking about are millions of billions of molecules. And if you are finding some molecule with good affinity properties to your protein, that's not the whole story. You need to optimize it also for other properties, because at the end, the molecule will be used in a clinical trial, so it's getting in contact with patients, so it needs to be less toxic. We need to have good distribution abilities. We need to produce it in our manufacturing facilities, so we need to have a good yield. So there are a lot of other things you need to take care of, which makes this process super complex.
And how does LLM is helping really?
Yeah, I can draw a picture here for this actually. And this is a picture of the needle in the haystack. But it's also not fully accurate. Why? Because the haystack underestimates the complexity here. It's actually a lot of haystacks and a lot of needles, and you need to find them. And the needle would be here the best molecule or the new therapeutic for a given disease. So you need to search them. How do you search them? As a chemist, you have a good idea how to search it because you are trained on it, but you are still a human. You cannot screen billions of compounds. It's just impossible. And this is actually where the LLM really helps you. The LLM, and specifically agentic AI, has the ability to find this needle by helping you searching it, by screening and narrowing down actually the potential molecules that are druggable and have desired abilities. And this is thanks to reasoning abilities of the LLMs. You notice when you are, for example, using ChatGPT at home, you can always ask ChatGPT, why do you get this answer? So there is a reasoning capabilities integrated. You have zero and multi-shot prediction capabilities. That means like large LLMs, you don't need to retrain them. You can use the context of the LLM and embed training data into this, and then trying that the model creates actually an output and a prediction, which means here the LLM knows the affinity based on the measurements that you did in your laboratory or on historical measurements that you can provide, so you don't need to retrain. It helps you searching fast in this space because you don't have this training process in place and you have something that is already usable and that you can instruct with human language. And this is one of the important things. It's like a chemical body that is not as good as you in chemistry when you're an expert, but it's very fast and it can give you some really creative ideas.
So if we had an example maybe that would make things even more concrete, I can see that it goes faster, not as expert as a chemist, but how does it work exactly?
Yeah, let me share you what we did. So in order to use, and this is the real innovation here, in order to use text-based language models for molecular design tasks, we teached actually the language models to be a small chemist. And this is based on this in context learning and agentic capabilities, which means like our large language models, you can talk to them and they can design compounds. And that's the real advantage. That means like you have really a small chemist body. But how you are interacting with somebody like this? Of course, you need to evolve the ChatGPT interface because it needs to be tailored to a chemist. So we did an evolution of this user interface by including a molecular draw editor and the possibility to upload molecules, as well also to interface with our chemical databases. So the LLM and the agentic framework sees a lot of the chemical compound series that we have in Sanofi. And that's it. You have a good chat interface, and then you can start actually the process. Let me give you an example, a concrete example how this works. And it's literally like this. Unfortunately, I cannot do a live demo. I would love to do a live demo here. So I did a live conversation snippet how you would use the application. And the cool thing is, it's as simple as it is. You are asking the LLM, what can you do? Similar like you would use Concierge at Sanofi. And it tells you what it can do. It can help you with molecular design search. It can help you with your reaction planning. It can help you with structural analysis. And it can help you also with the property predictions. And that's kind of a nice package that you need in order to search in this space. So the LLM shows you what it can do. And then you can directly interact with it. You can draw a molecule or structural compound that you find interesting. You can upload data, and then you can ask the model, hey, based on this interesting functionality and the data that I have in the small POC in the experiment, can you generate me new compounds that have also a high affinity to the target? And the LLM is doing this. And that's kind of a magic behind, because the LLM returns you a small space of molecules, also visualize the space of molecules, that you can see how similar they are. And then you can pick them, and this is the next nice thing that you can do, you can challenge the LLM. Similar like you can challenge the LLMs that you are using at home. When you are an expert, like a chemist, you can also say to the LLM, can you change something in the molecular structure that you predict? The LLM will give you an output of interesting structures, and you can say, and that's something I'm very proud of, we teach the LLM to do chemical modifications directly on the structure. It means like you can go to this interface and you can say, I want to change a functional group. Can you deprotect this group? And the model is doing this. And this works because, and that's a kind of a magic trick behind this, the magic trick is that the molecule itself is represented also as text in a very abstract form. As you can see, a small molecule is represented as this word, and the model itself can use this word to search for patterns actually. And also to use this to learn semantic similarities of the molecule itself. And that's kind of the trick. So it's a different modality, but somehow also in the text world.
And tell me, what makes it unique compared to traditional tools that we're using?
So what is here really unique, and that's the big innovation here, is that this is a conversational experience that enables a chemist, a medical chemist, to use AI without actually coding. That means like when you're working as a chemist in the laboratory and you want to do an AI project, you need to do this very classical. You need to talk to data scientists. Data scientists are building for you generative models for molecules, and then you're meeting them. You have a couple of project returns. You're iterating around the designs. You're parameterizing the pipelines. Long story short, you're working like 3 months, which is a long time because it's a typical project. And with the methodology and the app that we developed, you don't need to do this anymore as a chemist. You can directly iterate with the LLM on your generative designs, which means like you are having a faster process in creating the molecules. And on the other hand, and that's the thing that I really like about this approach, you are getting at the end, not faster the molecule, you are getting a better molecule. Think about the search space picture that I drew. Now you have the LLM that can search faster and you can search more, so you have a higher probability to finding a compound with better affinity, less toxicity, and a better production capability later in the factory. So all in all, it's a win-win-win situation that you have. And that's what we are doing for our patients, and that's the big innovation here. Like you're combining these two worlds, expert AI and generative AI.
And tell me, let's be honest here, we're putting in the hands of AI our health. So I suppose it's kind of at the same time a little bit, it could seem a little bit dangerous. How can we make sure it is really using it in a good way? Because we know about hallucinations now that we've all using ChatGPT. Tell me about that.
This is something that we really like in Sanofi, specifically working with AI and working responsibly with AI. And of course, I need to circle back. One important thing I need to note here, even though we are designing compounds with AI, we need to go to the same processes than any other pharmaceutical we are developing. That's kind of a thing. We are here in early research, so there's still a rigorous testing in vitro, in vivo, in clinical trials for the molecule. And also the government process, FDA, European Council, we need to go through all of these instances. And this is something we are also doing here when we're designing. The next point I have to say is, the AI does not work autonomous here. The AI is working together with a chemist, with an expert in the loop. So the AI is proposing things based on your feedback, but there will be always a human in the loop. When you are asking myself about the human in the loop concept, I think it's the best use of AI that you can have so far. Because if you are an expert, you know actually the topic and the commodity. In this example, when you are an expert, you are a chemist, you know actually the functional groups, the series, and what's needed actually. So you can challenge the LLM. You know what works and not. And the LLM just helps you to guide you through the process. But without your expert knowledge, you could not get to the molecule actually, because you cannot ask the right questions. This is similar when you're working with an LLM that helps you with code, when you are doing a Python program or something like this. Then you need to program. When you can program, you can challenge the LLM because you know exactly what it's doing wrong and where the hallucinations are. And lastly, and this is also based on the rigorous testing that we are doing and monitoring of our AI, in Sanofi we have the responsible AI as a very big program around all of our AI and governance. Which means, like, of course, we are checking with experts always if our AIs are ethical, fair, and also working on improving the accuracies of the models.
So it's very interesting the way you are actually using it, and we'll feel a little bit less stressed knowing that you are working hand in hand with AI, and without the one and the other one actually can't be as efficient. So let's watch probably a video that will summarize everything that you've checked.
We made it.
Introducing search and design in chemical spaces. Sanofi's revolutionary generative AI solution for early research in drug discovery. Imagine a ChatGPT-like interface that empowers chemists to create new molecules interactively and conversationally. That's exactly what we've built. This AI-powered tool tackles the challenge of designing small molecular compounds in vast chemical spaces. It transforms a process that once took weeks into mere minutes, bridging the gap between preparative medical chemists and computational experts. With features like in-context learning and human-in-the-loop design, our solution guides chemists towards innovative ideas, potentially leading to higher quality molecules. By simplifying workflows and accelerating compound development, we're not just saving time and costs, we're paving the way for faster drug development with fewer side effects. Search and design in chemical spaces embodies Sanofi's AI-first approach, empowering our scientists to deliver better outcomes for patients, faster than ever before.
Very interesting. It summarizes perfectly, actually, what we've said. Just one last question, and we will give the floor to you, dear audience, so that you can ask also your questions. What are the pros and the cons, I would say, of using generative models in scientific research?
Yeah, of course I'm biased, I have to say.
They're only pros, yeah?
I'm a data scientist. Of course, I have a longer pro list than con list, so I will start with the pro list. So as I told you, what is the best thing is actually you can see more molecules in the space. You have a higher probability to get better druggable candidates. You can reduce side effects a lot because you are doing a kind of a virtual screening, an LLM-guided screening, which is great. And at the end also leads to less animal experiments. So it's kind of a win-win thing because the LLM accelerates and creates higher quality. So generative models, generative design is specifically for small molecules, something super important and also used everywhere. But the good thing here is that it works really with a text-based interface. So on the other hand, and this is a cool thing about what I can say also as a pro, you have explainability also included. You have reasoning capabilities, you can create better molecules, and you can also record the way how you found this molecule. And that's kind of a really good thing because typically the design process, and it's always the problem of like your key scientists are leaving the company, of course a lot of competence is leaving the company, but if your algorithms and pipelines, they stay always in the company. And the knowledge is really abstracted there. So there are big pros, of course. And yeah, I need also to talk about a couple of cons. One con we already discussed here, and that's the hallucination topic. And that's, I think everybody knows this, because everybody's using generative AI, which is great. So you know the limitations. Generative AI, if you are asking myself, it's kind of a 90-10% thing. Like the generative AI and LLM models are like 90% accurate when you are prompting them well. So there is 10% missing. And that's kind of the thing. In order to use them wisely and accurately, you need to be an expert, specifically for expert tasks. When you're a programmer, you're using an LLM for coding, it's perfect, you are fast. If you're a chemist, you're using our LLM, you are also fast. But if you are not an expert, then it's difficult actually to use it and to challenge the model also for the hallucinations. Also talking about technical challenges. I showed you that the molecules are represented as these small words. Sometimes the LLM are breaking these words because the LLM understands the grammar of these molecules like by 90%. So it's not always guaranteed that the molecules that the LLM is generating are actually molecules that exist or can be cooked. So there's always also the synthesizability problem included. There are a lot of ways how to overcome this, but these are main challenges that you have when you want to use LLMs actually. One other challenge I need to, or big con I need to say, but on the other hand it's also a change in how we are working and how we are doing tech. When you are looking, for example, today at VivaTech, you see that the GTC is here, NVIDIA is here. Why is NVIDIA here? Because they are owning the graphic cards. People are talking about companies being GPU poor, GPU rich, because you need to have access to these big machines in order to train these models. And this is also super expensive. It's of course great for NVIDIA and their business, and you are seeing this also. But on the other hand, it means if you are a single researcher without funding, it's difficult actually to innovate and to develop an AI.
Great. Well, thank you very much. I think we can give a great round of applause to Sven. Thank you.
Thank you. Thank you, everybody.
Thank you. Do we have any questions in the audience? Both of you. No worries. And Sven, you're staying with us. Just
yeah, I'm staying here. No worries. I'm also afterwards here for questions.
Yeah, first, Sven, thank you for your amazing presentation because it made me feel smart. So it means it's a great presentation.
It's because you are smart.
I don't know. But my question is, if I understand clearly what you say, correct me if I'm wrong, when you're speaking about AI, you're speaking about augmented intelligence and not artificial intelligence, right?
We're speaking about artificial intelligence.
Yeah, but I mean,
as
a tool.
Yeah, exactly.
Augmenting the expertise of the scientist.
Exactly, yeah, as a tool for the scientist, actually, not an autonomous AI system, let's say it like this, yeah. There's of course also adaptive learning included in the algorithms because they can read your context and your previous conversations and take this to account to the new outcome that you have in your prompt.
And we have our second question over here.
Yeah, thank you for the talk. I have a question on your 90-10% hallucination.
That's a guess from my end.
Exactly. Did you actually test it, the results from the generative AI? And do you think that this 90-10 is true, even if it's complicated chemistry?
We need to sample, of course, more. I have this number actually on a paper that we published last year in ICML. So what you can see here, the model is based on a publication and an idea that we have. How can we actually change this language model to use many-shot in context learning for small molecular design? So we measured this. It was something like 90% of the molecules that were generated by our approach were accurate, and 10% were broken. So you can take this as a small proxy. But since the tool is new, and I'm also very grateful that we have so good collaboration partners from IDD in Sanofi that helping us doing the experiments, we need to evaluate it with more experiments. The results that we have so far are pretty great, where we are writing right now the second paper about this. But it needs to see what the future is bringing. But on the other hand, and that's also a cool thing I can mention here, the large language models are also evolving on the same time. It means like you can plug in a different engine into our approach, and so it will be always up to date. And that's kind of a cool advantage here. And maybe the 90% will be at a certain point 95 or 98 when GPT-5 is out or Claude 4.5. Depends, but yeah, thank you.
Any other question?
Hello, thank you for a presentation. I have a question. Is it a system that is in development, or is it already integrated to your processes of drug discovery? And if yes, did you succeed to measure the improvement in terms of discovery, research, what type of drug you succeeded to discover, and if you discover new one, is it extremely new one or is it adaptation of a previous one?
This is a very nice question. So on one hand, we put the application into production a couple of months ago, so it's pretty fresh. But before we put it into production, we actually did a big experimental series with our partners from IDD, that's the Integrated Drug Design team in Sanofi, where they gave us a couple of challenging molecular series to test actually the capabilities of the model to predict in a space that the model does not know. Can you transfer the knowledge from one molecular series to another? And that's what we evaluated. We have pretty nice results out, I have to say. We are writing a manuscript and hope that it's been published by the end of the year. So on one hand, making it short, we are on doing own research with the tool itself, checking actually how accurate it is. And on the other hand, we are also operationalizing it and making it accessible to more and more teams beyond IDD. And then I think in a year from now, we have a pretty good statistic actually, how well it works.
Well, thank you very much. Any other question? We still have 1 minute. Okay, one last question then, please.
Thank you very much. I just want to ask, please, how do you predict toxicity using SMILES configuration? Only 2D configuration? How do you...
We have actually two ways to do this, actually. So on one hand, it's an agentic approach, which means like there's an agentic node that can predict toxicity on classical models based on RDKit. And on the other hand, and that's the cool thing, the model itself has multi-shot learning capabilities. That means if you measure toxicity for a given organ or for a given assay, you can upload it and then you can use the model directly to be, I would not say trained, but to use actually your data in the context to do the prediction itself. We evaluated this in the paper that we did last year. It's it's not as good if you're training an own model with, let's say, a graph convolution neural network on a toxicity data set and then really evaluating it on an unseen data set. But it's good enough, actually, to use the understatement of toxicity in the generative process of the model.
Because for different molecules, we can have...
You have to speak a little louder.
Sorry, do you hear me now? Okay.
Yeah.
For different, you can have a different configuration for the same molecule. So for the same molecule, you can have... A molecule can be toxic and non-toxic.
Now I understand the question better. This is something we are covering right now, not fully. From the chirality of the molecules, there is a bit of understatement of the LLM, but I think in order to address this better, we need also to do far more prompting and learning on this. But that's a good point, yeah.
Thank you very much. A great applause to you again, Sven. Thank you.
Thank you, Christina. Always a pleasure speaking
Always
here.
a pleasure. Thank you very much. And please don't hesitate to visit our booth. We have virtual reality to discover the new facilities. We also have a GT3 car from McLaren. This car is actually racing on Saturday at Le Mans, 24 Heures du Mans. So go and check it. You will understand why on earth McLaren is partnering with Sanofi, and obviously a corridor from research and development up to the patient, how it goes. A lot of innovations as well, and startups. Enjoy, thank you very much, and see you in about 15 minutes for our next session on stage here. Thank you.